Hello everyone, welcome to our second Immunolab lecture. So today we'll be looking at enzyme-linked immunoassays, also known as enzyme-linked immunosorbent assays, or ELISAs, or enzyme-linked immunoassays, EIA. So essentially this is going to summarize applications for antibodies in the laboratory. So come back to this after the lecture if you'd like, but this gives us a hint into what we'll be talking about today. So, aka ELISAs, I'll refer to as ELISAs in most cases, but enzyme labeled, so in an ELISA, an enzyme labeled antibody is used to detect antigen or another antibody. So, for example, we have horseradish peroxidase and alkaline phosphatase. So, an enzyme substrate is converted to a colored end product, so usually immunofluorescent in nature, and this allows for quantitative measurement by comparison with a standard. So, we add an anti-A antibody covalently linked to the enzyme. So here we can see sample 1 is A, sample 2 is B. Then we wash away the unbound antibody. So these are usually done in wells with pipetting and stuff like that. So here we have our well now with antigen A. The enzyme makes colored product from the added colorless substrate. And then we measure the absorbance of light by colored product. And this is usually through machinery. So again, we use an enzyme substrate that is converted to a colored end product, allows for quantitative measurement, and is compared with standard. So again, here's another example. The well is coated with the antigen with an antigen. So usually, for example, we have antigen. So these coateds, usually you buy the plates usually. So I have a plate for IL-8, for example, and then or in either way, we have the antibody binding. So then we submit. So sorry, I got it backwards. Sorry about that. So we put our blood in, our saliva. At least from my experience, those are two mediums I've used. So our antigens will be in there. Then we add the antibody from the product. So these antibodies are made specifically to bind to, in this case, IL-8. And there's a ton for different cytokines. So let's just say we also have IL-6 in here. IL-6 will be in, these antigens will be in here. Let's just say if it's in the blood. These antibodies won't bind to IL-6. They'll wash away, but they'll bind to IL-8 as we intended. So we have indirect and direct ELISAs, but we'll talk indirect. Indirect ELISAs are used to measure concentrations of antibodies specific for a certain antigen. So how is this done? So we coat the wells of the plate with an antigen of interest, shown in the green circles. We add a sample to be tested for this presence of antibody specific for this antigen. So if antibody is present, this binds to the antigen. So we have a washing step. So this is when only antibody specific for the antigen on the plate remains. Then we add secondary antibody, which is conjugated to the enzyme, and this binds to the antibody that is attached to the antigen on the plate. So here we can see that sometimes they're called anti-antibodies, and these bind to the antibody that binds to the antigen. So then we wash and add the substrate, and then we measure the color change through optical density. So the diagnostic application of indirect ELISA. So for example, this can be used to detect antibodies in HIV. So wells of microtiter ELISA plates coated with recombinant GP120 for HIV for nonspecific binding. So further nonspecific protein binding is then blocked using blocking proteins. The serum is to be tested is added to the wells. If the antibodies to GP120 are present, they bind to the GP120 in the wells. These washing step removes any unbound antibody, and the antibody not specific for GP120 is washed away. So this works kind of different how I originally talked about it compared to IL-8. So instead of IL-8 being already here in the blood, so we're not testing the blood, we're testing for an antibody response. So now we have pre-coded antigens, and now we want to see if our body has antibodies. You can see we add the enzyme-linked antibody, and then we have this double binding again. So we have a detection step. So this is when the antibody specific for human antibodies is added. Anti-human immunoglobulin antibody, again, anti-antibody sometimes is referred to, are conjugated to the enzyme. So if anti-GP120 antibodies were bound to the GP120 on the plate, the detection antibody will bind, and this enzyme is immobilized on the plate. Then we have the washing step again, which removes any unbound antibody. Then we add a substrate, which is enzyme on bound antibody will react and causes color change. This measures the absorbance of the end product, and again, antibody not specific for the GP120 but washed away. We can see here, washed away, compared to the binding and the color change. So another example is this sandwich ELISA or capture ELISAs. So these are used to measure the concentration of antigen or desired substance like cytokines. So this is more what I was referring to earlier. So I kind of had it backwards earlier. So this is where we would have that IL-8 example I was talking about. Sorry about that. So this is done. So we coat the wells of the plate with antibody specific for the antigen of interest. Then we add our sample to be tested for the presence of antigen. So usually blood, serum, even sweat, urine, any kind of fluid. So if the antigen is present, it is captured by the antibodies on the plate. Then again, we have this washing step where only the antigen is captured by the antibody on the plate remains. Then we add our enzyme conjugated secondary antibody, which is also specific for the same antigen, but different epitope, which is very important. This binds to the antigen that is attached to the antibody on the plate. Then we wash and add substrate once again and measure the color change with optical density. So here we have our coated plate with the antibody. So again, this is IL-8, for example. Then we have the addition of the antigen. So let's say we add our blood. There's IL-8 in the blood. Here's IL-6, but this is irrelevant. Again, we have a control, which is no antigen. So we have remove it. These will both be washed away. We can see here we have the antibody, so we put blood in all three. But these are washed away, and again, these are not specific. No IL-8 will bind, therefore we won't see a color change. So here we have IL-8, that color change, and the binding of the antibodies. Another technique is the ELISA spot assay. So this assay can be adapted to measure cytokine secretion from cells by using a sandwich/capture ELISA format so that cells producing the cytokine of interest will produce colored spots. So this is kind of looking for what cells actually produce this compared to like the other one where we're just counting essentially the IL-8s that bound within that sample. Oops. So ELISA spot assays can be used to measure the production of antibodies by B cells. This is, again, we coat the wells of the plate with an antigen of interest. We add the cells to be tested, and there's a limited number per well, and we incubate and allow for antibody secretion. If antibody is produced, this binds to the antigen near the cell. The washing step removes any other antibody. We add the enzyme conjugated secondary antibody, and this binds to the antigen that is attached to the antibody that's attached to the antigen on the plate. Then we wash and add substrate, count and measure the spots that appear around the cells producing antibody of interest. So again, we essentially zoom in and count the spots, and then these spots are positive signals. So the ELISA spot assay can also be adapted to measure cytokine secretion from cells such as T cells. So we want to measure Th1 cells releasing interferon gamma, for example. So we can coat the wells of the plate with antibody specific for the cytokine of interest. So here we're talking about anti-IL-2. So we add cells to be tested with a limited number per well. So we add a bunch of T cells. Then we incubate them to allow for cytokine secretion. So if the cytokine is produced, it is bound by anti-cytokine antibody, so the anti-IL-2. And these coat the plate near the cell. Washing step removes any unbound cytokine. And we add enzyme conjugated secondary antibody, also specific for the cytokine of interest, and binds to the captured cytokine. Again, we wash and add the substrate, count and measure the spots around the cell to produce a cytokine of interest. So here we have a cytokine-specific antibodies are bound to the surface of the plastic well. Activated T cells are added to the well. These T cells are a mixture of cells with different effector functions. The cytokine is secreted by some activated T cells and captured by the bound antibody. Then the captured cytokine is revealed by a secondary cytokine-specific antibody, which is coupled to an enzyme, giving the rise to the spot of insoluble colored precipitate. You can see here. So you can see the blue binding to the other antibodies. So yeah, those are ELISAs. These are really commonly used in many studies, and a lot of the work I've done or will be doing in my research is looking at saliva or blood and cytokines, and this is used in a lot of exercise immunology, different stuff like that. But again, super relevant, and if you haven't had a chance to go into the lab and try this stuff, this is kind of a decent theory background, but there's also a lot of good YouTube videos out there if you want to, see the actual procedures done in person, and I recommend searching for it. But yeah, hope you learned something. Take care.